So today I would like to introduce our research regarding about the synthesis of bioactive nitrogen heterocycles. So this is my past and present research. Okay, I want to synthesize the bioactive from the nitrogen heterocycles. Okay, we start. Okay, so what actually is organic synthesis? Okay, from the moderator says my background actually master from the analytical chemistry and instrumentation. So my mini project actually on research of natural product. So once I further my PhD study, so I have no idea about the organic synthesis, because one of my team invited me to join research for the organic synthesis. So once I heard about the organic synthesis, I feel scared, because of organic synthesis. I imagine there are lots of molecule, compound, there are lots of reaction, and also you must strong in fundamental. So that is the specialty of the organic synthesis. So here, the organic synthesis, for your information, is a special branch of chemical synthesis and concerned with the intentional construction of organic compounds. So this is the process of making a desired compound by using several of chemical reaction. If you can see here, this is one example of the experimental that I've done with during my research PhD. Okay, so there are lots of glassware, and also there are very risk during handling this experimental. So that is the organic synthesis. It involve many of a chemical reaction. You must know the mechanism, and also you want know how to obtain the target molecule. Okay. So this is the framework from the natural product parts until our parts chemical synthesis or organic synthesis. So this is the pathway for discovery of drug research. Okay, starting examples for here, starting from the medicinal plant with the potential of activity, go through for the primary extraction, next for secondary extraction and fractionation. So produce a mixture of phytochemicals with a high possibility in biological activity. So what next? Okay, so this chemical synthesis play a main role part after the natural product is finished. Okay, means that we know the special biological activity in medicinal plant. After extract, get a mixture of phytochemicals and proceed for develop of a lead molecule with the higher potential in biological activity. So this is our part, which is chemical synthesis. We want to synthesize the lead molecule by using of a chemical, and also by using several or many steps of chemical reactions. Okay, from this part of chemical synthesis, so here I want to explain the chemical total synthesis means that from the natural product, apart natural sources, compound, and then we want to synthesize from the simple chemical precursor to produce the complex natural organic molecule or complex synthetic organic molecule via a different of a synthetic method or routes, and in some principles of chemical structure and also reactivity. Okay, once we obtain this complex natural organic molecule, we proceed for application. So application, especially in drugs discovery, we want to proceed for industrial, chemical industry or pharmaceutical industry. Okay, so total synthesis means that complete the chemical synthesis of complex organic molecule from simple structure, simple molecule, using commercially available of chemical. Okay, so this is the main purpose of organic synthesis, actually summarized from this picture diagram. So we construct of carbon skeleton by inserting of removal of functional group to produce a stable intermediate, and after that to obtain the product in a reasonable yield. So from the quantitative yield, so proceed for the next step application for science and technology area, especially in pharmaceutical, cosmeceutical, and also in chemical industry. So now, okay, so that is total synthesis. So I narrow my presentation. Okay, for my past and present research is about nitrogen heterocycles. Means that my target compound mostly from the nitrogen heterocycles. There are a lot of nitrogen heterocycles. So for your information, nitrogen heterocycle as a core structure in natural product. So means that in natural product, produce the quantity is very small, so it's not enough for the for industrial production. So that's why we need to do development in synthetic strategy, and also to scale up our amount of lead molecule with the higher potential by synthetic chemistry. Okay, so that's why a lot of the researcher do research about synthetic pathway of this ring nitrogen heterocycle, because most of the product market drugs containing at least one fragment of nitrogen heterocycles. About 80% of market drugs, okay, containing of the nitrogen heterocycles fragment in their drugs. So example drugs in pharmacy like Abilify, Actos, Bristol, Cymbalta, Nexium, Oxycontin. So all of this is the famous of in market drugs. Okay, so because of this feature, special feature of nitrogen heterocycles, so a lot of researcher want to synthesize this compound, because of want to improve the pharmacological, pharmacokinetic, toxicological, and physiochemical properties of drug candidates, and also ultimately drugs. So here is heterocycles in organic chemistry. Actually, what is heterocycle? Okay, means that a ring of connected atom. Okay, if you look here, ring, all these compound in a ring system. So in this ring, actually connected atom, where one or more of the atom in the ring are element different from the carbon. Okay, such as oxygen, okay, connect with carbon, okay, which is a different element in one ring system, and also the rest three nitrogen connecting with the atom carbon in one ring. So it means heterocycle means that a ring of connected atom where one or more atom in the ring are element different from carbon. So there are four category. Okay, so for the red circle, if you look here, so this ring connect with the oxygen. Okay, either one or more oxygen. Okay, carbon attached with this oxygen. Okay, for the blue circle, so nitrogen, okay, attach connect with the atom, okay, in the ring. So this one is my skeleton of my target compound for the for the blue circle. Okay, so for yellow circle means that sulfur connect with the carbon, and for the green is hetero. Hetero means that there are two different elements. Okay, connect with the carbon in ring system, one ring system. Okay, so there are several of heterocycle in organic chemistry, but for my past and present research, I'm only focused and I'm only interested for the blue circle nitrogen. Okay. So from the survey, most important heterocycle, if you can see here, okay, the bold one is very most important because of their many reports regarding the potential in biological activity. So here, example, pyrrole, imidazole, thiazole, thiophene, pyridine, pyrimidine, pyrazine, indole, purine, and pteridine. Okay, so it is the most important heterocycle because of containing the crude in biological activity or in pharmacological properties. Okay, so now I'm focused, okay, for my research past and present. Okay, actually I'm new in the research on organic synthesis. Okay, because from my experience from starting from the PhD and for the organic synthesis. So for my grant, I'm also work in organic synthesis from the natural sources compound. Okay, with the biological activity. Okay, for my PhD, okay, so I'm choosing pyrrolidine as a nitrogen heterocycles because of previous reported says that there are a lot of pharmacological properties in pyrrolidine ring system. So for your information, this pyrrolidine, which is an element connected with a carbon atom in one ring, okay, can divide it into three types of pyrrolidine, which is inserting by two carbonyl. Okay, so we call as pyrrolidine dione. So means that there are two ketone insert on the ring of the pyrrolidine. Okay, so if insert at position 2, 3, this one, this one is 2, 4 ring system, and this one is for 2, 5 ring system. So I'm only focused on system 2, 4 ring system because of that is my research in PhD. Okay, so my PhD should be entitled Chemical Studies Towards the Total Synthesis of Pachydermine and also bioactive metabolite derived from Chamonixia pachydermis. So means that from the natural parts, this plant is Chamonixia pachydermis only have in New Zealand. So after isolate, produce of degradation of pachydermine. So this pachydermine have the biological activity. So that's why I proceed my PhD, previous PhD, by using pachydermine as a target molecule, which is the main fragment unit is a pyrrolidine units. So for my present research, okay, so this is done by my master student, which is I'm focused on the nitrogen heterocycles indole, okay, indole ring system, which is want to get beta carboline as an intermediate, but my target compound is dibocarboline A. So title for my master student is synthesis of dibocarboline A derivative via tetrahydrobeta-carboline intermediate. Okay, so firstly, I want to sharing about my past research, PhD, which is I'm choosing the tetramic acid. Why? Because here is the biological activities of tetramic acid in natural product. Okay, there are lots of the products, there are lots of the molecule tetramic acid containing of the biological activities such as cylindramide. Okay, it's good for the tumor cell lines. Orontoside A as having a cytotoxic activity, at the same time it's inhibitor of a tumor cell. Okay, for hydrochloric acid, produce metabolite with an antifungal. Okay, deuterium cycline is a potent inhibitor of bacteria, cytotoxicity, hemolytic activity, and discordamide. It's good for the antifungal activity against Candida albicans. So this is some of example biological activity of tetramic acid in natural product. So what is my target compound is pachydermine. So here is my target compound, pachydermine. Okay, if you can see here, this pachydermine actually origin from New Zealand, Basidiomycin chamoisa pachydermines, from family Boletaceae. So here is from the natural sources, okay, natural plants, which is pachydermis, chamoisa pachydermis. Okay, it's like cendawan. Okay, so here is the fragment of the pachydermine molecule. Okay, this part is the main body of the pachydermine from tetramic acid. Okay, because this is a nitrogen heterocycles. Okay, at C3 position, inserting of the oxalic and at C5, we want to insert the 3-chloro-4-hydroxyphenyl substituent. Okay, actually this pachydermine is degradation product because of 5-3-chloro-4-hydroxybenzylated tetramic acid was reported there are a lot of the biological activity. So one of the report said that a mild antibacterial activity against Bacillus subtilis at concentration is 40 microgram per ml. So that's why I want to synthesize this molecule to know the variety of the biological activity of this derivative and also intermediate of pachydermine. Hopefully, we can display the similarity of the potential biological activity. Okay, so why I want to synthesize the pachydermine? Because there are no reports on the synthetic routes, and also because of the limited works on isolated compound of pachydermine and also its derivative. So that's why from this problem, so I have a novelty approach for the synthesis of the natural product pachydermine and also derivative via beta-beta diketoester. Okay, the other one, the problem is a previous study on such ring system contributed due to the high incidence of related biological activity, so overcome structural complexity of tetramic acid derivative. So because of this previous study, so I have done with a preliminary biological test on antibacterial study and also anticoron sensing, okay, to evaluate the potential pharmacological properties of all the derivative intermediate of pachydermine. So my objective for past research, okay, first I want to synthesize the co-skeleton, which is N-benzylated beta-beta diketoester. Okay, I want to synthesize this co-skeleton. Okay, and number two is I want to synthesize, okay, along the way to get the pachydermine. So there are several of steps, and of course several of steps produce a different of action and different of using different of reagent and produce a different of a derivative and intermediate towards to pachydermine. Okay, so here is a part for the objective 2. So number 3, objective for biological purpose. Okay, because I want to run to test all the derivative and also intermediate pachydermine to know the activity each of antibacterial and also antiviral. Okay, so for your information, my research is aligned with the SDG. We need that Sustainable Development Goals. Okay, so aligned with SDG number 3, good health and well-being. Okay, so it assists the current health issue, especially during COVID-19 pandemic. So it's a very important to develop the drug discovery. Okay, okay. So it's also a very give a high impact on society, economy, and also nation because of from the drug discovery and from the chemical synthesis part, we grow of the new chemical industry for the pharmaceutical and also medicinal manufacturing. Okay, so this is the framework how to produce the novel synthetic in our lab. Okay, from natural parts. Okay, natural product isolate from the plant, run tests on some biological activity, and after that, in our part organic synthesis, we start with the do some experimental in lab. Okay, with using a different of procedure, different of experiment, different reaction, and also different of static material. And then once we get the purification, after purification, we get the compound, pure compound, and run the instrument by using the instrument of NMR, IR, HRNS to confirm the compound. Okay, and after that, analyze the data from the instrumentation spectroscopy, and then proceed for the screening biological test. Okay, we do the some bioassay and focus on the publication, copyright, and also patent. Okay, so here is the framework of flow to produce our novel synthetic group or to produce the novel compound for organic synthesis part. So before we get the target molecule, okay, for organic synthesis, we start with to propose the synthetic route towards the packaging. Okay, of course, we start with the proposal, how to synthesize this molecule packaging. Okay, there are part A, which is we want, first objective, we want to get this beta-beta diketoester as core structure or skeleton from the starting material of glycine metal ester, okay, via condensation and decyclization. So we get the core skeleton, and after that we proceed for part B. Okay, for part B, we insert the acyl group, okay, by acylation. Then we want to insert the alkene functionality as C5, okay, by using the aldol condensation reaction. And then proceed for the decarboxylation because we want to remove this part of ester from this ring. So using decarboxylation, and after that, we want to hydrolyze ester to form H group. And last part, we want to remove the protecting group from N to get the target compound of academy. So this is the retrosynthesis to academy. Actually, there are 3 parts. Okay, because of academy can divide it into 3 parts. For part A is ketotautomer of academy, because this academy is a ring system, so it can be inverted in keto and imine form. Okay, so here inserting of the azalyl subunit and also C5 is aldehyde group. Okay, for part A, how to synthesize part A by using starting material glycine metal ester with metal malonyl potassium salt. You can get the intermediate of diester and proceed cyclization, cyclization to get or to manage the desired product. Okay, beta-beta diketoester. And then for part B also we synthesize this azalyl subunit. Okay, so it came from the starting material diethyl oxalate with potassium acetate, and then you get the octane or you get the desired of the substituent azalyl chloride here. Okay, so for part C is aldehyde, so you can buy from any brand of company chemical. Okay, because this is functionally available. Okay, so what I have done during my PhD student, this is my part. Okay, so I get a synthesis advanced intermediate of pachydermy in 6 steps. So this is my advanced intermediate of pachydermy towards to the pachydermy. okay, by using starting material ethyl iodoacetate. Even though from the proposal I'm using glycine methyl ester, but for the organ synthesis, we want to synthesize or we want to optimize the amount of the starting material so that we can produce, we can proceed for the next step of the reaction. Okay, you can do here. So this is organization, cyclization. Okay, this is my first objective to get beta-beta ester. Okay, insert at C3 position with acyl group. Okay, I have done. Okay, this is in recipe mixture. Okay, proceed for the C5. C5 inserting of the enzyme. Okay, but here I get the outer position, and then if I prolong the reaction, so maybe I get the alkene functionality at the C5 position. And then lastly for the carboxylation to remove the ester at the C3 position. So this is we call as an advanced intermediate of academy. And of course, there are several steps, 3 steps more to get my target compound academy. So this study, this research already published, okay, then already have the copyright for this one. Okay, so this one is I want to show the synthesis of the derivative pachydermine at C3 and also at C5 position. Okay, after I get the cosclitan, beta-beta diketoester, so I want to further my reaction with the several steps until I get my target compound. So all the way you get several of substituent, and also you produce different of derivative and also intermediate of pachydermine. So that's why in organic synthesis, the main objective you want to get target compound, but at the same time, along you get the target compound, you also produce a variety of the derivative and also intermediate of pachydermine. So your compound should be a lot of molecule you can get from the several steps. Okay, for my PhD is about 50 compounds. I have done to synthesize the compound by using a different of reaction. So here is my derivative and intermediate of pachydermine. Okay, so if you can see here, there are a lots of exploration of reaction. Okay, with a different of alternative routes to get my target compound pachydermine. Okay, actually my target compound not exactly same to the proposal, because I have done with the different of alternative route to get the best optimization and also to get my target compound. So after I've obtained all the molecule derivative of pachydermine, so I run for the biological test for all the derivative and intermediate of pachydermine. Okay, with antibacterial and also anticorrect sensing for my past research. Actually, I want to do more about I want to focus more about the biological activity for all my derivative and intermediate of pachydermine. Okay, so this is some antibacterial that I have done before. Okay, and also this one is for anticorrect sensing. Okay, this is to look or to evaluate their potential pharmacological properties. So here, show you the compound that show the antibacterial activity. Okay, so this one for the antibacterial activity and this one for the anti-correct sensing activity. Okay, after screening from the 50 compounds, so here about 1, 2, 3, 4, 5, 6, 7 is show some potential in biological activity for antibacterial and anticorrect sensing. Okay, so for the future plan drug discovery, okay, which is I need 3 more steps to proceed for my pachydermine. okay, which is you can see here hydrolysis. Sorry, I want to eliminate this one, okay, to form alkene functionality. Okay, so Ac3 I want to remove hydrolyze ester, and lastly to remove benzene. Then I get my target compound pegylated. Okay, so here is my publication and also my intellectual property for my work, research work. Okay, so I just summarize my past research. Okay, starting from the natural sources compound. So come up with the proposal, do some retrosynthetic routes, and done with the experimental. Okay, experimental of organic synthesis, and lastly around the biological activity. So all this process we call as the total synthesis in drug discovery. So here is the current research, okay, for my master student. Also from the nitrogen heterocycles, okay, choosing indole because there are lots of the biological activity and also pharmacological properties regarding indole nitrogen heterocycles. So here is my target compound, dibocarbolate A. So I actually isolated from the roots of Mulice dibocysts, exhibited anti-inflammatory activity by using inos with IC50 value of 18.41. So means that the smaller value of concentration, so will give the best biological activity for anti-inflammatory. Okay, so extracted from the stem of Mulice kedahensis. Okay, is done by Dr. Noor from campus UiTM Negeri Sembilan. So she is my partner for my grant. Okay, so I isolated only in trace amount. Okay, this is problem parts for the initial product. After isolate, and the compound shows the good biological activity, but the amount is very small. It's not enough for drug discovery. So that's why she passed their compound. So for me to design the synthetic approach, the dibocarbolate A. So this is my target compound. Okay, my master student. Okay, so actually the co-skeleton of the dibocarbolate A is beta-carbolene. If you can see, there are 3 ring system. Okay, and here is some example of the biological activity of beta-carbolene. Was reported. Tryptophan is good for treatmental mood problem. Pinoline is good for free radical scavenger, especially for antioxidant. Okay, and also for mind. Okay, here is an example of beta-carbolene. Okay, so this is some selected example of commercial drugs and bioactive molecule for beta-carbolene. So that's why I choose this nitrogen heterocycles indole as my project in my project research. Okay, so here is a proposed synthetic route I have done with my team members to synthesize the dibocarbolate A derivative by using the starting material 5-methoxy L-tryptophan. So if you can see here, there are lots of steps. Okay, so what my student have done during MCO, okay, because of the starting material 5-methoxy L-tryptophan is not arrived, so we're using the tryptamine to explore the step that I proposed before this. Okay, by using a different of starting material, but still in the same group of tryptophan. Okay, 3-ring system. Sorry, same group of the H indole. Okay, so here the derivative of dibocarbolate A by using the starting material tryptamine for 1 to 3 steps. So now we get more than 10 derivative. Okay, so after the chemical is arrived, so we proceed for our main route by using 5-hydroxyl tryptophan, okay, with a different of the glycol, okay, and we managed to get the skeleton, the intermediate of the beta catenin, that catenin A, okay, and then proceed for the reduction, and here is still on point, still on point, okay. So I want to show you that this is the synthetic route to get my target compound catenin A. Okay, so at the same time, I'm also in organic synthesis. We have many alternative routes, synthetic route. If route A is not successful, so we must come up with the route B. If route B is not successful, so we must come up with the alternative route C. So that's the organic synthesis, the journey of the organic synthesis. Okay, using a different static material, different fractions, so you can get a lot of the reaction mechanism, and also a different variety of derivative and analogs of the derivatability. Okay, so here is our Center of Chemical Synthesis and Polymer Technology. So this is our chemical synthesis group team, which is our head is Professor Dr. Ahmad Sazali Hamzah, which is he always encouraged me to do research, okay, regarding the organic synthesis. Not forget to Professor Dr. Zulina Shamiri, okay, my supervisor for this, and our team members, Associate Professor Dr. Muhammad Fazli, Associate Professor Dr. Nurhana, and Associate Professor Dr. Karima. So now I'm an associate member for this team. Okay. Also Associate Professor Dr. Muhammad. So here is the website for our team, for our center. Okay, so here is laboratory. In our laboratory, there are three labs. So organic synthesis research lab, where I have done my research past and present. Okay, at UiTM Punjab, and also polymer technology research lab, also at UiTM Punjab, and for microbiology research lab, tissue culture research lab at UiTM Punjab. Okay, so here is the organic synthesis research laboratory. Okay, this is our lab. It's during colloquium, it's during we received the invited speaker. Okay, this is some discussion or sharing session knowledge to the student, FRP, postgraduate. Okay, so here is our storage, here is our room for discussion, meeting room. Okay, so this is my arrangement. This is my postgraduate student present, master, Aina Bila Ashari. Actually, she registered on March 12th, still in progress. So this is some of activity we do in our lab. Okay, some recording video on how the experimental procedure done, and then discussion on during meeting progress, and the RP reference proposal. So thank you for listening my presentation today. Okay.
Thank you. So today, as a physicist, I could know that there are so many ring of atoms which relates to the cycles. Informative. So, Doctor, we are now moving towards the end of our session. So, can we have a couple of questions from the viewers?
Okay.
To all viewers, you can text in the chat box and I will read them up for you. No questions. Asking for attendance. Okay, but so I have question for you, Doctor.
Yeah.
So I'm very it was very interesting part for the for your research where it relates to us, basically, biological
Yeah.
part.
Yeah.
So what is your maybe your next plan for the biological part? What you plan to do?
Okay. So for organic synthesis part, so I have done before this the presentation. I need three more steps to get our target molecule. But for biological parts, which is I have done with the antibacterial and also antiquorum sensing. But for future plan, I want to explore more about the biological activity, not only antibacterial and antiquorum sensing, but I want to focus on cytotoxicity. Sometimes it's good for the properties of biological property is good, but the cytotoxicity is not. But it's not good for human, right? So that's why I want to focus on the screening cytotoxicity and the others of for pharmacological properties such as anti-inflammatory, because there are a lot of compounds. So we need to do more of screening on the compounds so that we know what is the biological properties for every of compound. Sometimes compound A, number 1 until 50, 1 until 10 is good for anti-bac. But if we run to other biological activity, so it shows that compound 30 to 40 is good. So that's why we need to explore more about their biological activity to get the novelty on the potential of biological. That's my planning for my future research. Progress research.
Understood. So we have a question from our viewer. So from Nurul Filzah, doctor.
Yeah.
So the question is, what is the percentage yield to synthesize the pachydermin?
Okay, so the percentage total yield for to get the pachydermin actually is about 3 or 4% overall yield. But this is need optimization actually. So we need times to obtain the large scale actually. Okay, so this is stage my research work during PhD. So maybe after this, I will focus to optimize the scale or yield to obtain the pachydermin. If the past research, I only get 3% for overall yield from step 1 until step 6, I get 3%. But after this, maybe I focus to optimize the every step until I get about 80% or 70% scale of the target compound.
All right, so there is another question from the viewer. I believe this is Dr. Sufri. Dr. Hidayah, is the pachydermin already on the market as a med?
Okay, Dr. Sophie, for pachydermin, actually there are no reports on synthetic roots on the pachydermin. So I'm sure on market drugs there are no commercialized for pachydermin compound.
Okay, doctor. So I guess that's all the question from the viewers. So there is no other question. Yeah. Okay, Dr. Hidayah, I would say that we have a pleasant session with you. It was very informative.
Thank you.
Thank you very
Thank
much
you. Yeah.
for your many success to come with your research
Okay.
work.
Okay, thank you. If any of participants are interested to do research in organic synthesis, total synthesis, then you can email me or you can come to our lab, IUS at Bioactive Pungot Alam, or my office at Bioactive Shah Alam. But during after all this issue COVID is settled, we can come and do some discussion about the synthesis. If you want to collaborate with me.
Okay, and thank you very much, doctor. So and thank you very much